So I wanted to try and do something a little bit different today. Rather than me talking and giving just a regular presentation, I'm going to make you guys do the work. I'm going to propose a problem. I'm going to impose a problem, and that is visual recognition, which is a question that I'm very interested in and really close to my heart. I'm going to introduce the problem, and then we're going to go around the table, and I'm going to force each one of you to ask a question. Imagine that you're a scientist and you're trying to understand how the brain works and how we can computationally model vision. What is it that you'd like to know? What are the experiments that you'd like to know? What are the what's the type of data that you need? What are the computational models that you will build, and so on? And sometimes the answer will be, more often than not, the answer will be, I don't know. Very often the answer may be, nobody knows. And in some cases, I may have something to say, and then I'll just expand on whatever it is that that you come up with. So first, I'd like to give a quick introduction about myself while you start sort of thinking about those questions. And don't worry, it's that there are no dumb questions. Any question is welcome. You are the scientist, and I'm just your Google assistant here. So hence the title, Everything You Always Wanted to Know About the Visual System, but Were Afraid to Ask. This is comes from a Woody Allen movie. I don't know if many people know about these. It's about sex, I think, not about the visual system, but that's what the movie was about. So a brief history about me first. As you can all tell, I have a very thick accent. I was born in Buenos Aires, Argentina. Growing up, different people come to science from different paths. Some people discover their passion for science late in life. In my case, I discovered that I was excited about being a scientist very, very early on. Many of you may not know about this. This was a pretty influential science outreach effort by an astronomer, Carl Sagan, and that was quite influential for me. My parents didn't allow me to watch too much TV, and this was one of the things that they did let me watch, and I was immediately fascinated, mesmerized. I saved money for a year, I bought that book, I read that book when I was 8 or 9, and then I decided I wanted to be an astronaut. My parents told me that was too dangerous, and so I changed from astronaut to astronomer, because they sound similar. And then I realized that what I really wanted to do is just be a scientist, and I ended up studying physics in the University of Buenos Aires. And then, being from Argentina, I spent most of my youth doing mathematics, studying physics, playing chess, and playing soccer not very well. I'm a really very bad soccer player. I usually joke about that. If you cannot play soccer in Argentina, they kick you out. So I left Argentina and I went for grad school to Caltech in California. And that was a fantastic transition for me. And that's when I started doing serious research. And at this point, I switched from sort of math and physics into neuroscience. That transition was very exciting to me. I was quite clueless about neuroscience when I started grad school. I find that most grad students, when they start these days, they are way more knowledgeable than I was at the time. I knew the brain was on top, the feet were in the bottom, and that was the extent of my knowledge. I was very confident with solving equations, with coding, all sorts of things, but I had basically zero knowledge about biology. And so the first year at Caltech, I spent time reading and talking to people and asking questions and going to seminars and whatnot. And that's how I started becoming a computational neurobiologist. From Caltech, I came here to MIT, where I did my postdoc. I was in Tommy Poggio's lab for my postdoc. And then after that, I went to Children's Hospital at the Harvard Medical School, where I have my lab now. And before I tell you more about what we do in our lab, I want to emphasize that all the work that we do is completely interdisciplinary, and depends on an amazing group of students and people. There have been many people coming by the lab, and I cannot do justice to all of them. This is just a partial list of many people that have come to the lab. People come to the lab from all sorts of different places. Again, this is not, it's neither updated nor exhaustive. This is just a handful. I'm still trying to figure out how to make this slide better. We just cannot fit everyone. But you know, people come from all over the world, and that's one of the things that, in addition to being passionate about science, that's one thing that I really enjoy, the possibility of interacting with amazingly smart people from all over the world, and that's something that I think is very unique to living in an academic world, and I always tell everybody, if I were a billionaire, and I'm not, I would wake up and I would do exactly the same thing every day, because I just love what I do. So just as a motivation, I talked about soccer. Soccer has been very important in my life, mostly not for playing, because as I said, I'm very bad, but in terms of watching soccer. But I want to illustrate, so we're very interested in questions about intelligence and how we can build intelligent machines. As part of the Center for Brain, Minds, and Machines, CBMM, that's one of our goals. We want to understand human and biological intelligence, and at the same time, we want to translate that into algorithms. And I'd like to illustrate with this how far we are in many ways. So let's see if this works. No, it doesn't work. OK. OK, I won't. It was working before. Not sure. OK. I'll show those videos before. What you were supposed to see is an amazing soccer player from Argentina called Lionel Messi doing all sorts of magic while he's playing soccer. And then this is sort of almost state of the art in terms of robotics and how robots play soccer these days. You can Google this, and it's kind of entertaining and fun to see how incredibly bad these robots are. Basically, there's a ball somewhere over here, and they walk 2 steps in a very clunky way. They fall every step, and so on. So I think the contrast between this and that is just enormous. Part of that has to do with things about building biological tissue and the agility of the human body and so on. But I would contend that that most of it has to do with the brain, with the amazing ability of the brain to do real-time, flexible computations in a handful of milliseconds. So the problem that I'd like to pose today for you to think about, I think I switched presentations. So the problem that I'd like to pose to you today is visual recognition. In general, we'd like to be able to take any picture like this one and be able to understand everything that's going on in there. And that's apparently, if you, I know that some of you are connoisseurs and have thought about visual recognition quite extensively, and some of you may be working in different fields. If you've never thought about vision and visual scene understanding, it seems quite trivial. You just open your eyes. Any young kid can look at this and understand pretty much everything that's going on in here. And yet I would contend that this is an extremely challenging problem from a computational standpoint. And we'd like to understand this at 3 levels. This is what David Marr, one of the fathers of studying vision, and Tommy Poggio refers to as the 3 levels of understanding. One level of understanding has to do with behavior. That is, what do you see? What do you recognize? What can you do? How fast can you do it? How do you move your eyes? All the questions about the behavioral aspects of how we understand a picture. The other level at which we'd like to understand this question is the neural level. We'd like to understand how neurons in our brains are actually responsible for deciphering what's going on in a picture like this. And finally, we want to understand this at the computational level. That is, we want to be able to write mathematics and translate that into computer code and have the machine see the world the way we do. That is, we want to teach machines how to see in a way that's able to explain human behavior, that's able to take into account the neurophysiological constraints. So one of the amazing things about looking at a picture is that we can ask essentially an infinite number of questions. Who are they? Where are they? What are they doing? You can ask questions such as, can you count how many shoes there are in that picture? I can ask you whether they are happy or not, who's looking at whom, and so on. This picture, even you can ask questions at even higher levels, such as what happened before, what do you think is going to happen next? This picture is supposed to be somewhat funny. You have Obama kind of playing a joke on this person here. You're probably all very young. I don't know if you can tell that this is a scale. You probably don't even know that they used to be very popular. This is how you, okay, so this is a scale. So in order to really fully understand this, you need to understand that people may be self-conscious about their weight. You need to understand that if he exerts a force here, he's going to change the weight. You need to understand that this guy does not know that about what he's doing. You need to understand that all the other ones do. That's why they're smiling. Perhaps they're smiling because it was Obama, and you have to smile because he's there anyway. But there's a huge level of there are multiple levels of understanding of a simple picture like this, and you'd like to ultimately be able to understand all of those levels, starting from the very basic questions such as what happens if I move my eyes in this picture, how do I understand that this is a shoe, that this is a hand, and so on, all the way to the highest levels of why is this a joke, or why is this interesting, or what's going on, that this is a reflection, and so on. So that's your task today. So I'm going to stop very soon, and I'm going to force each one of you to ask a question. Imagine that you're a scientist. You have infinite time, infinite resources, infinite amazing students, and you have to figure out how this works. You have to be able to do visual scene understanding. That's my imposition. I am imposing that question. So you cannot ask me about the olfactory system. The olfactory system is pretty cool, but that's the question that we're going to discuss today, okay? Within that, you can ask me anything you want, okay? And then I'm going to try to go around and ask the question. So imagine you're a scientist, and you say, well, what I really would like to do is an experiment where I do this and that, and that's my question, okay? So we normally think about how visual scene understanding works as a sequence of visual routines. The idea of a visual routine was coined by Shimon Ullman, and that's the idea of dividing and conquering. So we have a very complex problem understanding a scene. So we're going to try to divide that into a sequence of computational steps, each one of which may be much simpler to solve. For example, if we look at a scene, this is actually from an old, from a recent presentation that I gave. I just happened to be in Germany the day that they got out of they were eliminated from the World Cup, so I was just trying to make fun of them. But imagine that you have Obama's picture here, okay? So you have a scene, you have to understand that scene. First, you may want to extract a sensory map, just extract all the information that you can see from the picture. You may be able to propose an image gist. The gist of the image is something that you get very, very quickly. For example, you may know whether this is indoor, outdoors, whether this is an office or where you are. And then you may be able to propose foveal objects, that is the center of your fovea, what you're actually looking at. You may be able to say what's in there. This is the traditional domain of visual object recognition, and many of the computational models that we and many other people work on is trying to know what's in the fovea, what's the object that you're looking at. You may be able to make inferences from 1, 2, plus 3. You may have to do pattern completion. Some of the objects may be occluded, it may be hard to see, and you may have to put it all together. You may want to store that information in a visual buffer. There may be some task-dependent sampling that is there. Maybe some tasks that you have to do, maybe a question that I post about the picture. And then you're going to move your eyes. So all of this happens in about 200 milliseconds or so. And then you'll move your eyes to another location. And you will be scanning the picture, multiple locations. You will detect people. You will detect the spatial relationships, who's in front of whom, who's next to whom, and so on. And then these are routines in the sense that they are flexible. You can reuse them multiple times. You can repeat these steps multiple times. And then at the end of the day, assuming that there's a task, there's a question, you can come up with an answer. Each one of these steps may in turn have multiple subroutines. So one of the routines that we and many other people have studied more has to do with proposing what's in the center of your fovea, that is proposing foveal objects. So that function, that routine, may in turn query, this is jargon for multiple different visual areas, V1, V2, V4, these are different parts of visual cortex. So you may query those, you may integrate information from context, spatial context. You may select some sort of classifier to discriminate what's in there, upload that information to the classifier, and propose initial labels. So you can imagine that there are routines and subroutines and sub-subroutines and so on. And part of the question for us is to study those mechanisms for each one of those steps, for each one of those sub-steps, at the behavioral level, at the physiological level, and then at the computational level. So that's basically what I wanted to say. And then I want to turn this around and have you become the scientists who ask the questions and see if I can provide some of the answers. So it's going to be a different kind of presentation. So don't be shy. Unfortunately, you're sitting here, so you're going to be the first one. So I'm going to give you a few more minutes. OK. I want to say a few more things. And then I'm going to go around. And this is not an option. You have to ask a question. Okay, there's no bad question. There's no dumb question. Okay, so here are example questions. You don't have to ask these questions. Okay, this is just to give you an example and just to motivate. Okay, so we can ask me, for example, if I were a scientist and I were to study the visual system, the first thing that I would like to know is what happens when patients have lesions in visual cortex, because you study a system by breaking it up and seeing how it fails. Okay, so that's one question. You may ask, how do you study neuronal activity in cortex? How do you teach a computer how to see? So there are some questions that are very generic. I know that people come from different backgrounds. Some of you may ask very explicit questions. I don't know, maybe someone wants to know what is the role of parvalbumin interneurons in layers 2, 3 of anterior temporal cortex. I don't know, something super specific, okay? And that's fine, and we'll dig really, really deep into some of those questions. But it's also okay if you ask questions such as, how do you teach a computer how to see? Something that's very generic, okay? You had a question.
I was just asking if it had to be related to like scene understanding or just vision in general.
It's okay it has to be about vision in general, but you cannot ask me about whether there's life in other planets or about olfaction, right? So I want to restrict the question. Our goal is to be able to understand Obama's picture, okay? So it can be about vision, it can be about the retina, it can be about the inputs, about the statistics, anything that will help us understand at the computational, behavioral, and neurophysiological level what's happening in the brain. For the people who are moving to try to avoid asking questions or late, that's cheating. You cannot do that. No, I'm joking. I'm joking. No, that's okay. All right. We're actually going to start on this. That's okay. Okay. So each one of you will have to ask a question. Unfortunately, you're sitting here, so or fortunately, whatever. You're first. You're a scientist. You want to figure out how vision works. How do you do it? What do you want to know?
Okay, so if you look at like the Obama picture, for example, what happens when you see a specific face? How is that face like encoded in memory?
Okay, so how is that face encoded in memory? So let me start by showing this. This is a well-known diagram describing the architecture of the visual system in macaque monkeys. Each one of these boxes represents a brain area. Here we have retinal ganglion cells, so this is at the level of the eyes. This This is jargon for a place of the thalamus. This is called the lateral geniculate nucleus, the LGN. Information from here goes into primary visual cortex here in the back of our brains. And each of these boxes encompasses millions of neurons and a very complex internal subcircuitry. And we think that all of these areas, in some sense, are fundamental to our ability to do vision. I'm showing the macaque anatomical map because we just don't know this for the human brain. This is an amazing amount of work that people have done by injecting tracers and following connections between areas, and we just don't have enough information about this for the human brain. It turns out that there is an area somewhere in the inferior temporal cortex that is somewhere in this part of this diagram that seems to be essential for the ability to detect faces. And Nancy Kanwisher here at MIT did important experiments to study this area, and she refers to this area as the fusiform face area. The initial experiments that discovered this area were done by Charlie Gross and by Bob Desimone, also here at MIT, and then subsequent experiments by March Livingstone at Harvard. And it turns out that this area has a lot of neurons that differentiate between faces and other stimuli. And we think that it may be an important center necessary for visual recognition, partly because of lesion studies. And many of you may have heard about a condition called prosopagnosia. So if you have a lesion in this fusiform area that's responsible for detecting faces, people are essentially unable to detect faces. So it's hard to exactly describe because I suspect most of you are extremely good at detecting a face. There's a British neurologist slash communicator that passed away a few years ago, Oliver Sacks. He wrote a book entitled "The Man Who Mistook His Wife for a Hat." This is quite literal. This is a person with prosopagnosia who cannot distinguish his wife from a hat. So this is a pretty dramatic demonstration that there's something in that part of cortex that seems to be essential for detecting faces. People who have prosopagnosia may not even know that they have prosopagnosia. Now, this is pretty peculiar. I once happened to meet a very, very smart professor that had prosopagnosia, and he learned about this in the radio when he was in his 30s. So this person, he was very, very smart, so he could recognize people in all sorts of other ways. So he could understand people and recognize people by their gait, by the way they walk, by their voice, by their bodies, by the way they move, by all sorts of cues. He never realized he was not using face information. He thought that's the way everybody does it. And once on the radio, he heard about prosopagnosia, and he said, what is this thing about really recognizing faces? So people may function extremely well, even if you don't. But there's a particular part of the brain that seems to be responsible for storing the memories and information about faces. Carla.
I have like two questions. One of them might make you angry, and then the other one is like more like traditional.
Let's go for the second one then. No, okay.
I don't know. So I was reading one of your reviews where they talk about different lesions in the visual system, and there was a woman who can't can't describe objects, but she can reach for them and interact with objects, and then she can't tell orientations, but she can still like put an envelope in a mailbox. So like those people have like no awareness of their visual recognition, but they're still doing it. So is there like a cutting line for consciousness of vision versus like practical vision that you need to survive?
Is that the one that's supposed to make me angry?
Yeah.
Oh no, I'm not angry at all.
The real question is like, do we actually interact with the real world the way we think we do?
Oh, I see. So let me tell the story about this experiment that you are alluding to, so that everybody's on the same page. So roughly speaking, in this diagram, people tend to divide the visual system into two streams. This is the so-called ventral visual stream, and this part over here is the so-called dorsal visual stream. roughly speaking, the ventral visual stream is important for visual object recognition, recognizing shapes, recognizing colors, whereas the dorsal stream, this other part of the diagram, is more relevant to localize objects for actions, to detect motion, and to detect stereo. So more sort of spatial aspects of vision. This is a simplification. There's a lot of interaction between the two streams. They're not really as independent as I'm portraying them here. It turns out that there's one patient, actually maybe 2 or 3 by now, but usually these lesion studies are very rare, and so it's a very small n. But the one that Carla is talking about is a patient that had severe damage in the ventral visual cortex, particularly in inferior temporal cortex areas. So these people usually, there are 2 main types of conditions. So one is carbon monoxide intoxication, and the other one is encephalitis. And this can lead to infections or carbon monoxide poisoning that may lead to a pretty large lesion in temporal cortex. So this person had the dorsal cortex basically intact and major damage on this side. So first they thought that this person was essentially blind, and then they realized that this patient had some interesting residual vision. So the particular experiment that Carla was alluding to has to do with the following. They gave this patient an envelope. Imagine this is an envelope, and there's a slit through which he has to put that envelope. This is actually a pretty old experiment. In the old days, people used to write letters. There were envelopes. You used to put them through a slit in these places, and that's how people communicated with each other. No texting, etc. So this is a pretty old experiment. So you give this envelope to a person, and you ask this patient, "What is it?" And they say, "I don't know. I have no idea." So imagine you have your eyes closed, right? So that's basically he cannot see it. He cannot recognize it. What's the shape of this object? I don't know. He's completely clueless about what this object is, about the shape, and so on. Okay? And then there's this slit, and the slit through which he has to place the envelope can have different orientations. It can be like this, like this, like this. Okay? So if you ask normal people, they would grab the envelope, they would position it in the right way, and just put it, and it's a pretty simple task. So they asked this person to indicate with his hand what's the orientation of that slit. So you have to do this, this, this. Okay? Again, a pretty simple task. Everybody can do it very well. This patient is completely clueless. He cannot do it at all. Okay? So the orientation is this one, and he goes like this. So it's completely random. And then they say, well, here's the envelope. Just put it inside the slit. And the patient says, well, I cannot do it. I don't even know. Just do it. Okay? And it turns out that the patient can grab the envelope and put it through the slit quite well. So this is quite remarkable. This is somebody who has apparently no conscious understanding of visual shapes, and yet he can act on visual information relatively well. It's not as accurate as normal people, but still quite accurately. So based on that, on those types of studies, people have argued that this part of the brain, the ventral visual cortex, is relevant for conscious access to shape information. Whereas you can have visual action on this part of the brain. You can do a lot of visual action tasks, even if you are not conscious of that type of information. Okay?
So the splits between ventral and dorsal.
So this is a major split between the ventral and dorsal part of cortex. Okay? Let's go around, and then I'll give people the opportunity to ask a second question. Okay? All right.
So in the Obama picture, there are mirrors, and like me as a person can recognize that there's mirrors. So like the people that are in the mirrors are like twice in the picture. But how does something that's doing object recognition?
I'm not sure, so how do people know that?
Like how does something like something that's conducting object recognition recognize that?
That there are two?
Yeah.
Is that, okay. So. I don't know necessarily about, so let me start by saying that when you study the properties of neurons in visual cortex, one of the most fundamental properties of neurons is that they have a receptive field. That is, there's a particular chunk of the visual space that they are sensitive to. So all of the visual receptive field sizes are defined with respect to the place where you're fixating, what you're actually looking at. Okay, so right now I'm looking at you. So there may be a neuron that has a receptive field here. That means it responds to anything that happens in this part of the visual field. Okay, there may be another neuron that has a receptive field there, and so on. When I move my eyes, all of that changes, right? And so if I'm looking at Luke over there, then my neuron that was paying attention to this part now will be paying attention to, let's say, this part. Okay, so there's a particular location that the neuron is interested in, and those neurons tile all the space. So we have essentially a map of the entire visual field, with one exception, which is the blind spot. But except for the blind spot, we have coverage everywhere. Okay. So you could imagine here that you have some neurons. So let's say that you're fixating here. Okay, that's where your eyes are. There must be some neurons that have a receptive field here. Others have a receptive field here. Other neurons have a receptive field here. Others have a receptive field there. So you'd imagine that if there are neurons that are sensitive to faces, and more specific to Obama's face, you would have some sort of activation in this area, and those neurons would have a receptive field, and that type of selectivity would become activated. Now, if you're asking specifically about how you count things, then I don't know. So then that's sort of an interaction between vision and memory, and then you have to invoke a lot of other things. So let's say that you want to count how many shoes there are. Well, I suspect that what you would be doing is you would be moving your eyes, not randomly. You'd be searching for shoes in this case, okay? And we and many other people have done work on visual search. How do you move your eyes when you have a particular target in mind? And then once you detect the target object, you need to put that into some form of visual buffer and interact with some other part of the brain that's responsible for counting, which I don't know, and I don't think we have any idea of what parts of the brain are responsible for counting. But before we get to counting, there are lots of visual neurons that have receptive fields and have specificity for certain shapes and will allow us to discriminate objects in different locations. So we have all of the basic visual information, and then somehow that has to interact with the visual buffer, with memory, and with some other number abilities that allow us to count and so on. So there's I'm a vision-centric guy. I study vision, but of course, the brain is a whole, right? So this has to interact with a lot of other aspects of cognitive function. For example, numbers and counting. Okay.
So mine has to do with parameters. So human vision, there can be a lot of parameters, like what is the lighting, how far are they from the object, to where they can recognize it. How do you look at those types of things when you're programming like a certain visual recognition pattern?
So let me see if I understand. By parameters, so for example, you say that there's different lighting conditions. How do you recognize objects and there's different lighting conditions? Is that the question?
Humans,
yeah.
Like humans can do that. Like if it was dim in here, I would still know that there are chairs.
Right. So that's one of the fundamental challenges in visual recognition. It's very, very easy and completely useless to write computer code that will just memorize pixels. For example, you can take this picture, and I can crop this part here and just memorize those exact pixels. And then any picture of Obama that has exactly those pictures, I will be able to recognize it very well. But the problem of vision is that things don't look exactly the same thing. So you mentioned luminance, and that's one of the aspects. So if I were to lower luminance here or change the contrast in this picture and such transformation, you would still be able to recognize that. Same thing goes for scale. If you get closer to me, this would appear larger. If I occlude part of the image, you would still be able to recognize it. If I change the color, you can change the face viewpoint. You can change all sorts of transformations. You can make all sorts of transformations and change all sorts of things in the image, and you can still recognize the objects. So that's what people refer to as invariants in visual recognition, and that's one of the fundamental challenges. How do you build computer systems? How do you teach computers to see in a way that's very selective? So you want to be able to discriminate his face from his face, okay? Because that's fundamental to vision. But at the same time, you want to be able to be invariant to luminance as well as many other transformations. So how do you do that? maybe I'll get to more about computational models if people ask more about that. But one of the ways to do that is to build into the computational model some computations that are invariant to stimulus transformations. So to give you just one example, Tommy Poggio came up with a model a few years ago that was called HMax. And in this model, imagine that you have, I'll describe how you do position invariance, but the same idea can be applied to luminance and other transformations. Imagine that you're trying to recognize this object in this location or in this location. So you don't care whether it's here or here, you still want to be able to recognize it. So then you may imagine that you have neurons that are selective to this object here, other neurons that are selective to this object here, and then you connect those neurons to another neuron. This neuron will compute a maximum of whatever this neuron is saying and this neuron is saying. And by computing the maximum of the two, then this higher level neuron will respond to that object in a way that's selective because it's receiving input from these two. And it doesn't care where the object is. If it's here, the neuron responds. If it's here, the neuron also responds. That's the property of the maximum. It doesn't matter where the object is. Okay? So that's one simple way of building invariance to the position of the object. You could imagine that you have something like that also for scale, for example. If this is closer or farther away, you can compute a maximum across scales. And that's one way in which you can build invariant features that are useful for visual object recognition. But generally speaking, that's the fundamental challenge. That's the fundamental challenge in vision. How do you build algorithms that can generalize, that can recognize things in an invariant way? Because memorizing things is not useful. That's the central challenge, I would argue, in AI. When you're building intelligent systems, the most important question you always have to ask is, does the system generalize? If you build a model that does something, but it cannot extrapolate beyond what you have trained it for, then it's not that interesting. That's the difference in school when we tell kids there's a difference between rote memorization and actually learning to do something. When you actually know and understand, then you can apply those ideas to all kinds of problems. If you're just memorizing things, then you cannot extrapolate to other conditions. And that's the fundamental challenge for visual recognition. We may come back to this question as well. Okay? By the way, these are all excellent questions. So we need a dumb question. No, sorry, go ahead. No. I want to say these are all excellent questions. So this is great. Go ahead.
Okay, so I know visual processing happens on both sides of the brain, but what happens like if there are only lesions on one? Does the other half like compensate, or like what kind of deficits do you have? Like how does that change if it's further down in processing?
So this diagram, basically you have these, most of these you have it in the right hemisphere and in the left hemisphere. In most of these areas here, there's a contralateral mapping. So the right hemisphere maps everything that's on the left part of the visual field. Did I make it right? No. Okay. The right part of my brain maps onto everything on the left. Okay, and the left part of my brain maps onto everything on the right. So if there is a major lesion on my right hemisphere, then I would still be able to see everything that's on my right hemifield, that is on this part of the world, but not what's on this part of the world. So that applies to almost all this cortical area, everything after V1 here, except that at the very top, you have some neurons that have such large receptive fields that start to cross the midline and start to have information from both from both hemifields.
If the lesions are like in an area that does processing later, is that information still retained? So like if V1's not lesioned, like can the information that it processes still be used, or is it like it's just not used at all?
So for example, if you have a lesion in V4, can you still?
Like the information that V1 processes, is that still used, or like is it just like there's no connection between the two?
So largely, we think about this mostly in a sequential fashion, in a hierarchical fashion, such that this provides information here, this goes here, and so on. According to that model, if you have a lesion here, the information cannot propagate forward. That's not exactly true, because there are so-called bypass connections. So if you were to scrutinize this diagram and inspect it very, very closely, you would realize that V1 here, there are lines onto V2, but there are also direct projections onto other areas that bypass the next stage. So even if you have a lesion here, there are connections that bypass and go into the next stage. Those connections are typically weaker, and therefore you would still have a major visual impairment. So there is some residual vision. If you have a lesion here, there are still bypass connections, but what you can actually do visually is very, very poor if you have a lesion that completely takes out one of these blocks here. It's not zero. It's not zero because of these bypass connections. It's also non-zero because information can go here and then here. There are lots of other ways in which information can go through. But it's not good. If you miss one of those boxes, what you can do with vision is very, very limited.
So
Yeah.
that scenes have features that can be extracted, classified, and reused in a compositional and modular manner. How do an observer's priors influence information processing, and what systems are involved in this?
I'm sorry, say it again.
So I'm thinking that scenes
Yeah.
have information
Yeah.
that can be extracted and classified and reused in a compositional and
Yeah.
modular manner.
Yeah.
So how do an observer's priors, so prior information, impact the way that visual information is processed into, the relevant features that can then be reused to interpret another scene, or even to interpret and classify new objects that an observer has not used, and what systems are involved in this?
So again, we think that all this visual information is stored in here. Prior information is used in a lot of different ways. Let me describe a few of them. Okay, so a lot of the computational models of visual recognition that we have insist on this hierarchical idea that we have information going from here to here to here to here to here and so on. It turns out that in addition to these so-called bottom-up connections, there are major top-down connections essentially everywhere in here. So these lines are essentially bidirectional. The exception to that is the retina. There is no information going from the LGN to the retina. There are no back projections onto the retina. But everywhere else, there are top-down connections. So you may imagine then that any kind of prior information can be communicated in a top-down manner from higher areas onto lower areas in this type of diagram. There are lots of examples of how prior information can be very relevant for visual recognition. One of them is called priming. So you show some sort of a scene. Let's say I show you a scene of this of this room, and then I'm going to do object recognition. Then your prior so I'm going to show you an object. Maybe you're a prime for office-related or conference room-related types of objects, and it will take you longer to detect a picture of an elephant, for example. That's some sort of basic information that you have based on what you have seen. So that's sort of a short-scale type of prior information, and that's very, very likely communicated by these top-down connections. Another kind of prior information that you may be referring to is all your experience that you have throughout life with the visual world. So again, if you go back to this picture, it's not very easy to detect what this is. From your vantage viewpoint and with this projector and so on, you probably can see only a handful of pixels. But you have some conjecture about what kinds of things people may be holding in that kind of in this kind of scenario, right? So you may, even if you don't see it very well, you probably don't think that that's an ice cream or an elephant, right? So, how many things can it be, right? So there's a lot of prior knowledge that you have about the world that can help you interpret a visual scene. And generally speaking, and that's actually related to the project that Carl is doing, generally speaking, we don't have yet good models and good ideas about how to incorporate all of that extra knowledge. Some extra knowledge is incorporated in the form of correlations. For example, we know that maybe suits and ties usually go together, not necessarily, but there's some sort of correlation there. We know that maybe, I don't know what this is. Maybe this is a locker room. Maybe in a locker room, you have lockers and mirrors. Those two things kind of go together. And maybe lockers and elephants don't tend to coexist very often. There aren't that many pictures of, so that there's a lot of prior structure about the world that you know just because you exist and you've been here for quite a while, and you know all of this sort of structure about the world. And that prior information, I think it's very important. We still know very little about where in the brain, how it's incorporated, and how to use that in computational models to be able to capitalize on that for visual recognition. So lots of prior information at multiple different scales. Joey.
So I'm curious about how your brain decides where to saccade whenever you're presented in like a new scene, right? So you have no information about the scene, but you have to figure out the best way to search through the scene for the information. It's like a weird paradoxical situation that your eyes are constantly faced with. And so I know some people work on this, but I'm wondering if there are any like good models that can predict where people will saccade given a new scene.
Let me show you one. So today we just got an email about a paper that's accepted about visual about predicting eye movement. So I have to show this. But generally speaking, let me start by saying that. So I'm going to show you a couple of pictures now about a visual search task. So I ask you to search for an object, and that influences your eye movements. And I'm going to show you a quick sketch of a model to answer your question about how that works. Before I do that, let me say that if you're not doing visual search, I present you with a random scene. There is no task. There's nothing that I ask you to do. There are computational models that people have used to predict eye movements. These are mostly bottom-up saliency algorithms, and they work relatively well. And those are based on things like contrast. One of the most important things is movement. So if you look at a scene and something is moving, that immediately grabs your attention, and people tend to saccade to that. If there is a yellow car in the midst of the city, that immediately attracts attention. So this is just basic contrast in the image, color changes, motion changes, anything that's a change, a temporal change or a spatial change. So for example, if I'm presented with this scene with a picture of this over here, my eyes will probably not fixate on this location here because it's not that interesting. There isn't a lot of contrast here. But then there's a lot of other things that show sharp spatial or temporal changes. Okay?
One follow-up,
yeah.
Because I don't know like the proper distinction, but there are like saccades that like you perceive like when I look around this room, right? But then there are also like micro saccades, right? That happen so quickly that we don't actually even register them. And so do people study those as
yes.
well? Okay.
I'm talking, so everything I'm talking about right now is saccades. So the difference between a saccade and a microsaccade is the size and the temporal scale, as you said. So people also study microsaccades. It's much harder to study microsaccades. We have a lot of very good tools to measure eye movements. Nowadays, measuring eye movements is pretty standard. So if you have a person, you can measure, in a visual system, we mostly speak about visual degrees. Degrees of visual angle. Roughly speaking, the size of your thumb, if you extend your arm, the size of your thumb is 1 degree. So a saccade encompasses typically multiple degrees of visual angle. Let's say anywhere from 2, 3, all the way to 10 degrees of visual angle. So it's a movement, let's say, from here to here to here. Those are the kind of eye movements that people make. In addition to those saccades, as you alluded to, there are microsaccades. Even if I'm fixating, I think I'm not moving my eyes. It turns out that if you pay close attention, people are constantly moving your eyes. Your eyes are never still. You're constantly making very, very small eye movements. And that you can also study. To a first approximation, I think it's safe to kind of ignore that, in the sense that that doesn't seem to change our visual object recognition capabilities very much. Some people are now starting to say that that's not quite true, that even those very small eye movements may not be random. But to a first approximation, we can say that they're largely random.
Yeah, I think my initial question is more targeted towards saccades.
So most of what we and many other people study are the large saccades. One more anecdotal and intriguing comment about the microsaccades, those very small eye movements. I have not done this experiment myself, but several people have replicated this. So it turns out that if you prevent those microsaccades, if you don't let people move the eyes very, very quickly, the visual world disappears. That is, everything vanishes. And this is very, very curious, because how can the world disappear? I mean, it's pretty counterintuitive. So these are very hard experiments to do. Basically, what you have to do is move the world very quickly when people move their eyes so that it's completely perfectly in sync with those eye movements. So people think that those very small eye movements are essentially there so that our brain doesn't get bored, that there's no adaptation, and that we can constantly see the world. So our ability to actually see anything depends on those very small microsaccades. So without those microsaccades, when I flash a picture, you would see that picture for 100 to 200 milliseconds, and then things will essentially fade. And that's pretty curious, I think, in and of itself. But anyway, so most of what we and many other people study are saccades. How do you move your eyes in a scene? And then there's these bottom-up components that have nothing to do with tasks. And then we recently did a series of studies trying to look at visual search experiments. So now there's a scene, but you're actually trying to find a particular object in that scene. So these are 3 different experiments. I'm not going to go into details about all of them, but the basic idea is that there's some sort of target object. This is the target cue, and then there's some sort of scene, and you're trying to look for that object within that scene by moving your eyes. This is very, very small for you, but this was actually presenting in a full screen. So you have a full screen, and you're moving your eyes until you can actually find that object. Okay. We did the same thing with isolated objects, and then just for fun, we did it also with the Waldo, the famous finding Waldo experiments. So I just wanted to show this to give you a flavor for a computational model that addresses some of the questions that were asked earlier about invariance, but also that can reproduce to a pretty good degree eye movements. So here's what this model does. So this model takes a picture and processes that picture through a cascade of basically bottom-up computations. For the aficionados, this is a deep convolutional network. More specifically, this thing here is VGG16. This is a particular computer vision model that has been very successful in object recognition. So in the computer vision community and in the computational neuroscience community, people build models like this. Each one of these boxes contains many units. And then you can have a picture, and you can go through all of these computations here, and you can train the weights in that model, for example, to do recognition. So there are competitions. These are the kind of things that computer vision compete about. So you have a collection of lots of pictures. Each one of those pictures has a label, for example, a chair, a table, and so on. And then you have lots of pictures, you have lots of labels, and you can train the weights, the connections between units in this model, such that the model can thrive in putting labels to objects. So what the visual search model does is you take a target object, you're going to look for that object. I'm sorry this is so small. Trust me, I'm a scientist. There's an object there. So this is an object. You're supposed to look for that. Imagine that that's Waldo, for example. So it goes through your visual system, and then we store that information. So we think of all of this as ventral visual cortex, as a chunk of the brain that stores that visual information. That's what I was alluding to before. And then we store that at the top. I'm not tall enough to reach there, but it says prefrontal cortex. So we have a visual buffer, a working memory buffer that stores information about what you're looking for. And then you have a search image. This is where you're supposed to look for the object by moving your eyes. So then again, this is processed through the same ventral visual cortex, through VGG16. And then what we do here is that we have top-down modulation. These are those top-down connections that I was referring to earlier. So those provide modulation from prefrontal cortex on through visual cortex to create an attentional map. So this is an attention map that basically has information about how similar the different locations in the image are to the actual target. And this attention map will say that the location that's more similar to the target object, to what you're looking for, will dominate, and that's where the model will move the eyes. So the model will select the maximum of this map. There's a winner-take-all mechanism. We select that location. The model moves its eyes to that location. And then if the target is there, that's great. We moved our eyes. We finished visual search. We succeeded. That's great. If the target is not present here, then we have infinite inhibition of return. That is, in this case, the memory, the model has infinite memory. We don't want to keep going back to the same location, even though that's the maximum of the attentional map. So we want to select the next maximum. And that's how the model will move eyes to one location and the other. And this is a pretty reasonable model that can capture a lot of the variance in terms of how humans do visual search. For example, here, what this figure shows is the fixation number and the cumulative performance. So again, people have a target image, there's a search image, the subjects will move their eyes, and we're measuring how likely is it that they will find the target in one fixation, about 30%. In 5 fixations, it's about 90%. In 6 fixations, it's pretty close to 1. For humans, it's not exactly 1. Humans do not have infinite inhibition of return. They do go back to the same locations. So the red line here is humans. The blue line is our model. So our model provides a pretty decent approximation to understand how people move their eyes during these type of visual search tasks. And this is all task dependent. If you don't have the task, if you just have a picture and you ask people to examine the picture, then you have much less influence from these top-down signals, and then eye movements are largely, but not completely, dictated by bottom-up saliency, contrast, color, motion, and all of these types of features. Your turn.
So my question is sort of related to Josephina's question, where so like I was looking, right, we were looking at the picture with Obama, and basically I was thinking, like, you can recognize a picture, but also, like, there are also parts of the picture that mean something to you. So I'm very interested in, like emotion and the limbic system. So I guess, like, to put it simply, my question is, have you tried thinking about how the structures of the limbic system, including the hippocampus, well, I think especially the hippocampus, relate to or communicate with parts of the cortex?
So in this diagram here, if I can find it, so one of the problems of doing this is that I waste a lot of time switching slides. But anyway, so in this diagram, the Fellman-Van diagram, it turns out that over there, that's the hippocampus. At the very top, the pinnacle of the system is the hippocampus. So, and you'll see that there are lots of lines that go into there. So the highest echelons of visual cortex send their information to the hippocampus. I'm very interested in the hippocampus. I would contend, however, that you don't need the hippocampus to do visual recognition, that you can do very well without it. And the reason I say that is, again, going back to lesion studies, there are famous lesion studies in the hippocampus of patients, like the most famous one probably is someone called HM. So this patient had epilepsy, and many years ago, some of these seizures were originating in the hippocampus, and then to treat the patient for epilepsy, what the neurosurgeons did was remove the hippocampus bilaterally. So they removed the hippocampus in the right hemisphere and the left hemisphere. So this patient, HM, and many other similar patients with medial temporal lobe lesions, with lesions in the hippocampus and other areas, seems to be able to see very well. He can understand the Obama picture very well. They have severe long-term memory deficits. if you've seen Nemo, this is the Dory, I think is called one of the fish there. It's the paradigmatic example of short-term memory without long-term memory. That's a classical example of what a patient with bilateral medial temporal lobe lesions look like. Go ahead.
I guess, like, yeah, you could be able to see, but also there's an encoding of meaning, like, there's an encoding of valence that parts of the limbic system is responsible for. And if somebody can't, if they're able to see, they are able to identify objects, that's one thing. But I think having the ability to assign meaning to something will allow a person to function normally and to experience emotional states that'll motivate like certain behaviors that are maybe normal. So, yeah.
So, in a sort of caricature fashion, the amygdala seems to be quite critical for emotional processing and for valence. And again, it's not actually shown, but Nessan they decided not to put it in this diagram. But the amygdala would be also over there at the top, and also has strong connections to the visual system. So I think there's a lot more to cognition in the brain than what's shown in here. I put it here. There's the rest of the brain, right? So there's a lot more that needs to happen. But I would still contend, to the best of my knowledge, patients who have no hippocampus can still understand the meaning of an object, can still understand that that's a scale, that that's Obama. All of those, I think most of the questions that I posed over there, who are they, what are they doing, all those sorts of things, my understanding is that they would be able to answer those questions very, very well. They may never remember that they saw that picture. If I show them the picture again in 15 minutes, they would think that they see the picture for the very first time. They may not know that Obama is the president, because for that you need to have some sort of memory. There are lots of things that they may not be able to do. But if I ask the question of, is there a mirror in this picture, or tell me how many shoes there are in this picture, my understanding is that they would be able to do that very well without the hippocampus. Now, the amygdala is a slightly different story. So the patients who have lesions in the amygdala may have deficits in interpreting and expressing different aspects of emotional valence. But I would contend that even in those cases, they can still see and recognize objects and understand. So a lot of the answers to those questions, I think people will be able to do very, very well. It's not to say that the hippocampus is not interesting or important. I have spent a very large fraction of my career thinking about the hippocampus, recording data from the hippocampus. I really like the hippocampus, but I think that strictly speaking, for some of the questions we're talking about today, I think it's not needed. Yeah.
Okay, so my question doesn't have as much to do with the Obama picture, but if you could pull the soccer picture up again. Okay, so when someone looks at a picture like that, they can infer like the trajectory of the different objects in the picture, or in this case like the people. Like they have an idea of like what motion is taking place like before and after this moment was taken, or I mean at least I do. So like do you think that's something that is happening as like a prefrontal cortex, like taking in the context and figuring that out and like assigning that, or do you think that some of that information is taken from like area like MT and like the dorsal stream, even though there's not actually motion happening, like kind of processing what motion would be happening? Like is that all context and figured out after the fact, or something that like would be like picked up in the initial like?
So I think most of the answer to your questions I don't know. Let me start by the things that I do know, and you probably know these things too, but let me just state them for everyone. So when you look not at a picture, but actually at a video, or in a case where there is motion, there's temporal information in the real world. There's a particular part of the brain that's responsible for detecting motion. That's area MT, also known as area V5. And this area is critical for our ability to discriminate that things are moving to the left or moving to the right, and so on. Patients who have lesions in area MT or B5 have a condition called it's called kinematopsia, I believe it is. And the way they usually describe this condition is if you go to a disco and you're dancing and you have those stroboscopic lights, you know, when they basically have very strong flashes of light and then darkness. You see people dancing, you see them like this, and then you see them like that, but you don't see the trajectory. So that's what it feels like to not have this motion detection area. You see the world in snapshots. They say that, for example, you're pouring water on a cup, and because you don't see the trajectory, most of these people essentially end up with water all over, because they see the cup, they see the water, but they don't see the whole trajectory, so they don't know exactly at what moment to stop it. So that's the sort of what we know about motion. Now, what I think you're getting at is, so going beyond that, even in a static picture, you can make inferences about what happened before and what happened next. And those inferences you can make at different time scales. So maybe you think that this guy raised his hands. I don't know. That's a sort of a short time scale. And I don't know where that happens. I don't know if that requires an interaction between, in the real world, we don't quite see just that, right? So we actually would see the whole thing. So we have the temporal context information. We do have MT activated and sending information to the ventral stream and so on. In a static picture, I would conjecture that maybe you'd also this may also depend on those interactions, but I don't really know. And then maybe, I don't know if that's what you're asking, but maybe you're asking about things that in my world, they go way beyond vision, right? And I know even less about the answer, right? So you can make inferences about longer time scales, right? This guy is going to celebrate, this guy is not, right? And I don't know, that sort of thing. These are really high level aspects that you can grasp from an image. That go way beyond sort of the basic aspects of vision and notion and so on, and which I think are fascinating, but I have no idea where in the brain, how in the brain, and what are the computational mechanisms that give rise to that. But if you're talking about the inference that this guy probably raised his hands right before this picture, I don't know. I would conjecture that this may have been an interaction between these motion-sensitive areas and the ventral stream recognition areas.
I've kind of the opposite side of Becca's questioning. As far as there are obviously things I could pull out from an image that are not just image recognition. I can get humor and social context and all these other things that happen in other places in the brain besides just the vision center. So when we're building models of the visual system, can we only do object recognition, or are there some like mid-level questions we can answer, like does this object belong with this object, or is this whole like thought process kind of irrelevant because it's so isolated, doesn't have any connections to anything else like it does in the brain, so it, you know?
So I think we're just scratching the surface of what we need to. We need to understand I'd like to understand everything about vision. I'd like to understand not only the fact that these are shoes, but also the proximity of different objects, the interactions between objects, and all of those things. We understand significantly less about those. As we go to higher and higher level questions about an image, our understanding at the detailed neuronal and computational level decreases quite sharply. I think those questions are all fascinating. As you said, there are social interactions happening here, and those might influence how we visually interpret the image. In addition to social interactions, you mentioned, I forgot what it was. So humor, I think is a very, very, very challenging one. So in a few weeks, I'm going to be organizing a summer course, and we have projects in the summer course in Woods Hole. You should all apply to come to the course next year. It's a lot of fun. But we have summer projects. One of the projects that I want to do is be able to have a computer system that will detect humor. So I'm trying to get rid of language, because language is sort of another domain. Just visual humor. So I give you 2 pictures, okay, or I give you 1 picture, and I have ratings of whether people find it funny or not. Imagine some pictures are funny, some pictures are not funny. I think we're extremely far. I think that's super challenging. Even understanding these type of humor here, I think it's super, super complicated. I honestly have no idea where to begin to think about building a system. Imagine that if these few pixels are here, then this is not funny anymore, right? So there's so much that you need to understand to really capture humor. I think we're still very, very far. I'm not trying to be pessimistic. On the contrary, I'm trying to be optimistic. I think you guys are young, and just there's so much that we still need to understand about these problems. Even if you have a visual system that can label shoes, hands, I imagine that we can understand visual occlusion. I think we may be able to say this leg is in front of that leg. I think these are things that I could conjecture computational systems that may be able to do that. Having a system that will understand whether a picture is and predict humor, human ratings for humor, I think that's super, super challenging. So.
Intuitively, we would need like a model of the whole brain to do that, or maybe just like the vision system and like some humor system or some social system.
Not the whole brain. So let me start by saying I'm always very unfair to the cerebellum. I don't know if you know, but the cerebellum is a huge chunk of the brain, lots and lots. To a good approximation, I think that we don't need to model the cerebellum to understand this. So it's not the whole brain. I would conjecture that we don't need motor cortex. I think even without motor cortex, you can understand. So, but I think that it's way beyond our model of the ventral visual cortex. So it's not the whole brain, but it's way more than what we have right now. So at the very least, you need to understand that this is a scale. You need to understand that this guy is not aware of what's going on. There's a lot of stuff that you need to understand about the world in order to be able to decipher that. And all of those questions, high-level cognitive questions, I think we're still very, very far from a detailed neuronal understanding of the mechanisms of what goes on in there.
So I had a question before, but now I have a newer question based on what you were saying. So, but isn't, like, with humor, it's relative? So it's kind of determined of your own experiences, because someone's humor could be very different from another person's humor. So that's the, like, I think that would be extremely difficult to be able to have a computer system to, like, determine what is funny and what is not. So how would you, like, approach that if you could have, like, in what sense? I'm still trying to formulate the question from what you were saying.
Right. So to a very good approximation, I would argue that a lot of what we're studying here is pretty universal. So yes, people have different backgrounds and different education and different exposure to the visual world, and maybe some people come from Australia and other people come from Africa, and some people study math and some people study psychology. But I would say that most of us would probably give the same answers to. So I think there's some universality to a lot of questions in vision. So I'm pretty, but as you start to get into questions like humor and other things, then things start to, there's more individual variation. And I agree with you. So ultimately, we'll have to capture that. It's about who you are, and some jokes are funny because of the context, and otherwise they're meaningless, right? So there's certainly a lot of interesting questions about individuality. I don't work in humor, so I don't really know, but I would guess that there is some sort of universality to humor as well. I bet you that we can come up with, I don't know about this one, but I bet you that we can come up with pictures that 95% of people find give a rating of 8 or more in a funniness scale, and other pictures that people give a rating of 2 or less, let's say. So I think we would be able to operationally define some sort of universality of humor. So that's the project I wanted to do for summer. Take a lot of pictures that don't have any text, and then have people rate them, okay? And if 8 out of 10 people say that this is funny, well, I call that somewhat funny. And if 8 out of 10 people say it's not, I think there's something relatively universal, but of course I agree with you. There's a lot of individual variation, and ultimately, if you want to build models, you have to encompass that. But even just the universal aspect of humor, I think that's still already super hard, even without worrying about the fact that we're all different and find different things funny. But
you also think goes hand in hand with, like, intuitive physics as well, because, like, if you see, you have that person and they're on a weight, then you're like, they have this specific mass, and then, Obama is pressing on it, making the mass, like, go up and you're like, oh, my intuition. You know what I'm trying to say? I'm trying to like get it out.
I think I'm not 100% sure what Josh means by intuitive physics. I think there's a lot of things that you can put in that bag. For example, I bet you that if this person were levitating 2 inches from the ground, that would violate the laws of physics, but you would still understand the picture very well. You would still be able to answer all of those questions very well. So I'm not sure that's too relevant. But for sure, in order to understand what's going on here, you need to understand that he's exerting a force. And if you don't understand that he's exerting a force on the scale, you're never going to understand what's going on here. So there are some aspects of, So I think there's a lot about high-level cognition that's happening in here. Intuitive physics, I think, is a very generic term that has a lot of fascinating aspects, and some of that may be super important for certain questions. And I think that's the exciting things about what Josh is really looking at. For example, is this going to fall or not? This is the classical question, and I think that's fascinating. I'm not sure that you need to understand physics to be able to answer who are they. I bet you that if I violate physics here, you can still answer the question, who are they? But again, it's not black or white, right? So it depends on the question. And clearly, we have a lot of power to decipher whether this is going to fall or not. That's very clear. And I think that's a fundamental aspect of certain questions and certain aspects of visual scene understanding, for sure.
Yeah. So my question is about. I guess like historically, there's been a lot of like fitting really simple linear models of neurons, like tuning curves or patches, like receptive field estimates, like you mentioned. And to me, that seems at odds with like the new trends in modeling, which is to fit really like crazy nonlinear deep nets that are not interpretable to neurons instead. So how do you kind of reconcile these two approaches, or how do you see like the historical things that we do know about simple tuning of neurons informing modeling going forward?
So I think that's a great question. So let me just to put everybody on the same space. There has been 5 decades or so of studying neurons by putting electrodes and studying certain neurons that have preference for certain orientations. For example, that's the Nobel Prize-winning discovery of Hubel and Wiesel. There are neurons that are sensitive to curvatures. Then there are some people that say there are neurons that respond to faces more than to chairs, and all sorts of tuning properties of individual neurons. And that has sort of inspired and informed everything we understand about function in the visual system. And yet, as you say, many of the current models don't quite fit into that. So my conjecture is that we'll have to make major revisions to how we understand what neurons like. I think we have been quite biased in our understanding and exploration of macaque and even human neurophysiology. So one of the problems is that imagine that you're recording from a neuron. You have an electrode, you insert in the brain, you're studying the activity of a neuron, and you want to ask the question, what does the neuron really like? What is that really excites that neuron? So we cannot, it's essentially impossible with current techniques, you cannot exhaustively sample the stimulus space. If you imagine that, let's restrict the visual world to 10 by 10 pixels, and each pixel is binary, black and white. Even that's already basically have 2 possibilities times 10 by 10 pixels, that's 2 to the 100. That's already more pictures that you can show to any neuron that we're recording from. Of course, the world is not 10 by 10 pixels, so if you think about 1,000 by 1,000 pixels, and you think about at least 256 colors, or if not 256 times 3, it becomes, we cannot exhaustively sample. So what do you do? Well, most of the visual system has been done in a pretty ad hoc, hand-picked way. So people started showing patches of light. Then Hubel and Wiesel serendipitously realized that orientation was important. And then somebody says, well, let's start showing objects like these and see what happens. And then Bob Desimon and Charles DeGrosse, one day, they were walking in front of the monkey and they realized that the neurons were going like crazy. And that's how they discovered face cells. Because when they were in front of the monkey, the neurons would would activate like crazy, and when they were showing all the real stuff that they were actually studying, the neurons could not care about anything they were doing. But whenever they showed up, the neurons would start firing, and that gave rise to the whole study of face neurons. But I suspect that we'll have to revisit that in a major way. And let me very quickly allude to work that we're doing now along those lines. So we're trying to let the neurons dictate what they like by coming up with. I'm going to go through this very quickly, but I'm happy to discuss this in more detail. Basically, we have a computational model, a deep convolutional network model, that's a generative deep network. This is generating images. So we generate a bunch of images. We record the activity of a neuron. When I say we, I mean Marge Livingstone at Harvard. She's the one doing the monkey physiology experiments. So she's recording the activity of a neuron. And then based on the activity of the neuron, she's saying, I'm going to discard a lot of these stimuli that are not very efficient. I'm going to pick the strongest ones. And based on those, I'm going to go back to the model, generate a new set of stimuli, and so on. So I'm going to build a deep generative model that's dictated by the neural preference. And this is one of those experiments. We start from noise, and we come up with something like this. Okay? And eventually we find tuning that doesn't quite fit into any of the traditional ways of describing the world. So my guess is that if we do this kind of thing and we go in an unbiased way to explore neural tuning, we'll have to revisit a lot of our notions about how neurons are tuned in cortex.
How can you train this model? Like, don't you need tons of samples from that neuron to?
So there's no real training here. So the model was trained for visual object recognition. So this whole model is pre-trained. And then the neuronal part is basically a loss function from one generation to the next. So we can do experiments for 1 hour, 2 hours, 3 hours. And that's what we're doing to evolve. So this is evolution of pictures that are exciting for a neuron. And what we've seen is that some of these pictures that we evolve are much better than the thousands of faces and chairs and tables and orientations and things that we and many other people have been using for decades. The generative process was already pre-trained. We're not retraining anything here. So all of the weights in this generative process are fixed. Okay. I want to go through. I'm happy to discuss this in more detail and even send you what we're writing up on this. Okay. But I want to go through the rest. Go ahead.
So a couple of months I saw a talk from this researcher called David Eagleman. He works with sensory substitution, and he actually developed a vest that people, like deaf people, could use to hear words. So basically, the vest would vibrate into differently depending on the word, and they would kind of know what they were hearing. So I'm wondering, is there anything, with the knowledge we have of the visual system, is there anything similar we could do for the visual system, or do we not know enough yet?
So I think in principle one might be able. So this is one idea that's sort of starting to emerge in the vision community, which I still think is pretty premature, but quite exciting. Imagine for a second. Imagine a world where we solve computer vision. Okay. Imagine that given any picture. This is a picture by Sandro Botticelli, Allegory of Spring. Imagine that you have any picture and you have some computational model. For the aficionados, AlexNet, VGG16, your favorite computational model. Imagine that this computational model can answer any question you want about the image. We're not quite there yet, but imagine that you can detect Obama, you can detect shoes, you can detect interactions, maybe not quite humor yet, but a lot of understanding of what's going on. So now imagine that you have somebody who's blind, okay? How do we go from computer vision to a blind person? How can we convey that information to this person? So then there are a couple of options. One that people are trying is visual prosthetics. So really going into the retina or visual cortex and stimulating. And that's good, and that might work, and that's very exciting. One of the problems is how many channels you have. So you have very few channels. Another thing that people are trying is sensory substitution. And they do this in sometimes weird ways. Some people are doing electrical stimulation of your tongue. So they try to stimulate the tongue in blind people. Okay, imagine that I want to convey different pixels, and you have electrodes in your tongue, and you have sensation in your tongue that's meant to represent the visual world. It turns out that you can train blind people to do this. They hate it, or but it's doable. It's a way of doing sensory substitution. It's basically similar to what you were discussing. But the other idea that the simpler version of this is, what if we have this system, we could somehow even verbally dictate to people what's going on, right? So I can ask you to close your eyes, and I can walk around with you, and I can tell you, well, look, be careful, you're about to bump into these. Be careful what you say, because so and so is there, and it's a beautiful day today. The flowers are blooming. I can describe the world to you, right? So the problem with auditory inputs and language is that it's very slow. An image is worth 1,000 words, right? So if I want to describe what I'm seeing here and describe everything I'm seeing, it's very so this part I think is still pretty challenging. So this is where maybe sensor substitution, cortical prosthesis, verbal inputs, language. There are lots of ideas. I think this will happen. Exactly how, this is still an area of active research. If people are interested, there's a lot to do here. There's a lot to do here as well. This is where most of us work, trying to build better and better models. But if you have very good models, how can we help people who are visually impaired, or maybe even blind people? And I think that's doable. I think we're on the verge of helping them. But we don't have the right ideas and technology. We need smart people that will come up with better ways to do this. Yes?
When we search through a scene, we have kind of an intentional spotlight, and this is to help us get through a scene faster, know what we're looking for. And we often look at things but don't notice things if we're not looking for that. So when does that kind of processing stop? So we can see certain things, but it may not travel as far to like where we recognize what it is. So where does that kind of stop in the brain, and also computationally, when things don't need to be processed to save time or resources, how is that decided?
So if you're asking in the particular context of visual search, that's absolutely right. So you can move your eyes over something and still not visually recognize it. If you've ever played the Where's Waldo game, I can guarantee we actually have evidence for that. It happens all the time. It's very sad. People are actually looking at Waldo and they don't realize, and they keep moving their eyes and keep searching for Waldo. There are other notable videos, funny videos. Maybe you've seen the one with the gorilla and others, where there are huge things that happen in the world and you just don't notice because you're not paying attention. At some level, I think that's at the heart of the question about visual consciousness. When are you conscious of what you're seeing? And that's still not very clear. There are certain correlates of consciousness. We think, for example, that the retina is necessary to send information, but it doesn't reflect the contents of consciousness. For example, the retina can do a lot of things that we're not conscious of. For example, when you're watching TV, every frame is shown at a certain frequency. Our retina goes crazy. Every time there's a frame, the retina starts firing. Mostly you don't notice that. You don't notice frames, you actually notice a narrative, a story, a continuum, right? So we think that our contents of consciousness are not in the retina. We think that they are higher up, and people still fight about, is it V4, is it IT, is it a combination of the two, is it a combination of those, the activity there in other areas, and so on. So there's a lot of active research trying to understand the neuronal correlates of consciousness. That is, which particular neuronal firing patterns correlate with your moment-to-moment consciousness. So if you're looking for Waldo, your eyes may be there, you may process that information, it's in the retina, it's in V1, but somehow it doesn't quite reach your ability to say, yes, I got it. So computationally, we have very crude ways of modeling that. We have a visual recognition system. So I told you we move our eyes, and then we do visual recognition. If we recognize it, that's it. The visual search ends. If we don't, we keep searching, and then we can come back to that location. But there's very clear behavioral data. As you pointed out, people do come back to the same location over and over. They miss the target many times, and clearly there is something more to vision than just focusing on that. Then you actually have to explicitly attend to that location or to that object, be aware of what you're seeing. Yeah.
So my question is related to orthostasis. So I know that with orthostasis you can either go from color blindness
to atypical visual loss, to complete blindness, and the person doesn't notice because the mind creates these scenes based on previous images, previous experiences, and it just creates this like real life world, and the person just keeps on going, just doesn't never notice that he or she is blind. So what I wanted to know, based on Luke's questions, if that person feels like he's seeing something, does his neurons, like visual face recognition neurons fire? Can you just sit a person down and do the same eye tracking study on them or something?
I don't know. I haven't worked with this type of patient. But let me say something simpler that happens to all of us. If you cover one eye and you just look at the world, you think that you're seeing everything, right? It turns out that there's one location that's the blind spot. The blind spot is essentially where all the retinal ganglion cells leave the retina. There's a part of the visual field that's not covered. When you're looking with both eyes open, it turns out that the other eye compensates. So you have that information in the other eye. But if you're looking at the world with only one eye, if you close one eye and you look at the world, it turns out that there's a hole. There's a place in the visual field, and there are lots of demos online where you can convince yourself that this is true, right? So you can type demo visual blind spot online and just see. So there's a whole place in the visual field that there's nothing there. There is no visual information, and you don't see a hole. And the reason is that we invent what's happening in that region. That's called filling in. Basically, the neurons around that region are sending information to that area, and part of our visual world is just a construction. So there's a small part of the visual world that's my blind spot, and I don't see black in there. Okay, and that's because everything else is sort of filling in and completing what's in there. And in that case, people have done recordings, and people do show that in higher visual cortex, there are neurons that respond to the content in that location, even though there was no visual input at the level of the retina. So this is a very small aspect of your question. So yes, we do make up stuff. We do fill in and make up stuff, and there are neurons that are responsible for that. I do not know about this patient. This is a much more extreme version. Another extreme version is visual imagery. I ask you to close your eyes and imagine an elephant, right? I don't know why I'm coming up with so many examples of elephants today, but I ask you to think about an elephant, okay? So something must be happening in your brain to create this imagery of an elephant. And we and many other people have tried to study that, but we still don't know very well. There have to be neurons. There are neurons that respond to this visual imagery, even though there's no visual input whatsoever. So even though the retina is completely silent, you can create in a top-down fashion those types of activity. Other examples are hallucinations and so on. But I mean, in terms of this particular patient, I don't know. I don't know if people have done those experiments.
My question has to do with how does the encoding of visual information, how is it affected, or is it affected by different physiological states, like emotional and positive valence, or hunger or anxiety? I imagine that object recognition is fairly robust, but downstream visual information, how it's encoded in a more long-term way. Do we know how that might be affected by physiology or different emotional states? And if so, in what kind of areas? I imagine it's pretty large analysis.
You can construct scenarios where if you're hungry or not, or sad or happy and so on, you may visually interpret certain images in different ways. Most of what we and other people have worked on, I think, is, again, back to this idea of universal vision. My sort of null hypothesis is that most of these questions, you would have answered the same thing before or after lunch. And if I'm cruel and I don't let you eat for 2 days, my guess is that you would answer these questions in exactly the same way. I don't think that being hungry or not would affect a lot of what's happening in terms of visual processing. Now, you can argue that, well, when you get to humor, well, I don't know, that may be dependent on your mood today and whether you find something funny or not. You could argue that there are certain images, maybe you're in the desert and there's something that looks like water, and it's not, there's some sort of mirage that looks, right? So there are ways in which I think the state, and there's many conditions where a lot of internal properties and states can influence visual recognition. But I would say that there's some sort of core visual machinery where a lot of these we can study it in a way that's rather independent of many of these other conditions to a reasonable first approximation. Often in science we make approximations, right? We try to delimit things at least to start studying a problem, and then once we know more about the problem, then we can start sort of building upon that. But we think that we have a lot to be entertained even with this universal basic, without having to worry about some.
I have kind of more of a molecular question. So, like, RGCs don't have the ability to regenerate. What do we currently know about why that is? Slash, like, what sort of molecular mechanisms do we know, and are we looking at, for like example, like treating like glaucoma, are responsible for like axonal growth?
So I'm not an expert on this, so unfortunately I cannot really answer this very well. I do know that there are some of our colleagues that are working on exactly this question. It's a very hot topic in vision, and they do experiments in animals where they try to cut the optic nerve and try to use different types of molecular cues to get the axons to regenerate and to grow. You may know more about this. Let me say a few sort of very basic things that I know about. There are certain molecular chemical gradients that dictate where axons go, and that dictate under what conditions the axons will elongate or not. There are critical periods for that, and if you ask me to name specific molecules, I can't. I have very poor memory. But people have been working and making a lot of progress in trying to elucidate the molecular code by which axons grow. Why does a certain axon go to V1, whereas another one goes to A1. A1 is the auditory counterpart of V1. So there are chemical signals like traffic lights, if you will, that dictate axons will go one way or the other. And again, maybe I'm preaching to convert. Maybe you know this more than I do. But people are working very, very hard on these developmental questions and looking for what kind of molecules and so on. Why is it that the retinal ganglion cells in adults typically will not regenerate, whereas you have other species where you can cut the brain in two and it will continue to regenerate and so on? That's a fundamental question in the field of regeneration. People are working very actively on trying to find what's the magic of the chemical, what are the chemical molecules that will reactivate the regeneration process. And I have several colleagues that live and die for that question.
Is more into emotion as well, because I'm interested in emotion. So how do we give like a visual stimuli, emotional significance? So for example, faces contain like information about our internal states and we make facial expressions. So one thing, but not just face. Maybe the context could also be important. So how do we like give an emotional significance to a visual stimulus?
So roughly speaking, in the brain, I mentioned before the amygdala, which seems to be very important in processing emotional information. There are now, in the computer vision domain, there are now a lot of people who are interested in this question about whether you can recognize emotions in pictures. And you can treat this in the same way that we treat visual object recognition problems. So I mentioned this computational model VGG16, where you have a hierarchy of computations, you extract features, and then you train all those weights to say, this is a chair, this is a table. You could imagine doing the same thing, but now instead of saying this is a chair, this is a table, you can say this person is happy or sad. And people are doing a lot of these computer vision applications to try to be able to recognize emotion. So in terms of purely facial information, my sense is that this is going to be extremely successful. Now you mentioned context, and I think then the problem explodes in complexity, because often it's not just the facial information is not just one thing, but being able to incorporate a lot of different contextual information. That's partly again back to what Carla is working on with us. And I think that we have a lot to do still to understand how contextual information will influence emotions and so on. Again, most of what people are doing right now in terms of computer models, you have a hierarchical model, you have a lot of weights, you have lots of pictures that represent, say, happy, a lot of pictures that represent sad, and you train your model. And that approach works reasonably well for discriminating, let's say, happy versus sad in faces, for example.
but there was like a study that was done with tennis players that were either very happy or very sad, but people were not able to discriminate just because they were looking at the faces. So I guess like the context is very important. To have a context, your experience is also important. To have, you know, to have like this prior state. So like if a person has not experienced that, has any experience with a particular state, and is supposed to classify like a visual situation as like, this is sad. For me, I was supporting Germany, so I was like sad about what some other people. So emotion can vary, and it's very difficult to do.
Absolutely, I know that the pictures that you're referring to, and I think that's a beautiful example of how we will not be able to purely do this based on just facial pixel information. I agree with you. There's the temporal information, there's the temporal context, there's the spatial context, and we will have. Again, this is not exactly what I'm working on, but I think that we will, in the same way that we're starting to think about a lot of these questions for basic visual recognition, for understanding emotions, we need to incorporate all of that. And again. Plenty of things to do. So people who are interested in this, lots of opportunities to incorporate temporal and spatial contextual information to understand emotional valence.
If you were to build like a computational model from scratch to try to understand images, do you think like it would just naturally converge to like mimic the brain, or do you think like there's something about like the visual system in our brain that's like inefficient or like suboptimal?
So I guess we would have to define exactly what we mean by optimality. My guess is that soon we'll have visual recognition systems that are way better than we are. There's nothing special about our visual system. So again, we'll have to be careful about how we define better. Let's start with something that's happening already now. Barcodes. That's probably not very interesting to you, but computers are way better than we are at visually recognizing a barcode and interpreting it. Probably that's not what we evolved for. You can make lots of arguments about why, but that's a visual domain that's already, computers are better than us. Computers are getting better than radiologists are discriminating, for example, is this picture, does it contain a tumor or not? I think radiology will, as we know it today, will disappear. So that's another visual recognition domain. I think computers will get extremely good at face recognition. We're kind of there already, but not. But not quite. So I think that we can do it much better. But then you can start incorporating a lot of other constraints. Our brains are not just doing tumor yes or no, or Joe versus Susan. That's not all we do. We do way more than that. Okay. Not only that, but we do it with a constrained size, right? We cannot just, and also we do it with very little energy. So, you know, I don't know how much you ate, but you know, you eat. We can give you one plate of food, and you can do all sorts of things. Computers right now need enormous amounts of power. Another thing that's amazing about our brains is that they are very robust. So you kill some neurons in all sorts of ways, and people still function and see very well. If you take this computer and you remove some of the transistors, not good. It's not going to continue working, right? So if you start incorporating all of these other constraints, I think there's something fundamentally powerful and interesting. You use the word optimal. I'm not sure exactly how to define optimal, but there's something very, very profound about biology, about millions of years of evolution. But we didn't just evolve to scan, to understand barcodes, right? But if you're asking, can we build machines that will be better than humans in basic visual object recognition tasks? Yes, absolutely. I think in some cases we're there already. In some cases we'll be there very soon. We'll trust machines for many visual recognition tasks much more than we trust our eyes. In the same way that we don't trust humans to calculate the square root of 7, right? But I think these days probably very few people know how to actually do it, right? And we trust computers. I think in vision the same thing will happen. But again, as we start going to further and further questions about valence, about humor, I think there's still a long way to go. Okay. We're almost done now. I want to open it for round 2 of the questions, or if anybody has any other fundamental question. Yes?
How do you think that adding recurrence is going to change our models of visual systems? Like having
Recurrent connections. Yes. So this is alluding to, so a lot of our computational models right now essentially are bottom up. The information flows in only one direction. We and many other people have been building models where you add recurrent connections. So a couple of comments about recurrent connections. One is that, as you probably know, strictly speaking, you can take any model that has recurrent connections and expand it into a larger feed-forward model. So there's nothing really magical about recurrent connection. So if I give you a model that has recurrent connection, I can take that recurrent connection and just create another layer with weight sharing that does the same thing. So strictly speaking, I think that's not going to radically change the computation. However, there are a couple of reasons why it may be very interesting to do things using recurrent connections. One of them, perhaps not as exciting to you, but perhaps very important for biology, is again going back to these considerations of things like size. We cannot have a brain that's the size of a house. Biologically, that would just not work. It wouldn't work because of size. It wouldn't work because of locomotion, because of energy. So size matters. And being able to do computations with fewer neurons and fewer weights, biologically, that's a fundamental constraint. Maybe Google doesn't care. Maybe you don't care. But if you're thinking about biology, that's a fundamental constraint. The other thing that's, I think, very attractive about recurrent connections is that you can flexibly decide on the spot how many computations you want to do. And that may be something very useful for vision. If I tell you that you're going to do 20 recurrent computations, you can always expand that into 20 more layers. But that's not very efficient, because then you always have to do 20 layers. Maybe there are certain problems in vision that you can do in 100 milliseconds, others you can solve in 150 milliseconds, others in 200 milliseconds. And in some cases, you want to use one recurrent loop, in the other case 2, in the other case 3. And you can flexibly decide how many computations you need for any given problem with the same architecture. You don't have to change the architecture. You cannot have to say, oh, I'm going to wear this brain to ask who are they, and I'm going to wear a different brain to ask what are they doing, right? Or so you can't do that, right? So the same architecture needs to be able to flexibly solve multiple different questions, and that's something that I think is very appealing about recurrent connections. But if you're just saying I want to discriminate chair versus table, and you build your fancy recurrent connection, recurrent network, somebody can come and say, well, I'm just going to convert that into a feed-forward network that mathematically does the same thing. But any more questions? Nope.
I'm trying to find it. So if you have matched behavior to a net, let's say you have a net that behaves the same way, or in the case of the visual search model, it behaves the same way. You've matched the behavior, but then it's still kind of mysterious, right? Exactly what's happening in the brain. So you have somewhere to move forward. So like, what will your next experiments be, like after that work? So you've matched behavior. Now, how do you know that what the net is doing, that you've dictated to the net, is actually what the brain is doing?
So I think ultimately matching behavior is interesting, but not sufficient. As a silly example, you can build a model that predicts how a car will navigate and understand absolutely nothing about mechanics. If you don't open the hood, you'll never understand how a car works. Even though you may be able to predict, there's a red light, the car will stop. And you may have a perfect predictor, unless you are in Argentina or some places where people don't care about red lights. You can have a perfect predictor of behavior, of how the car will behave, and understand nothing about the carburetor, about the mechanics, about gas, about all sorts of. So if you really want to understand the mechanisms, there's no way around. We have to open the hood. We need to open the brain and look at neurons. So in terms of computational models that try to explain biology, we need to record from neurons. We need to record the activity of neurons during the same tasks, and then match the models to neurophysiological data and compare the two side by side.
So would you record from somebody while they're doing the visual search task in different visual areas, and then try to...
And then ask, can I predict the activity of the neurons from my models? For example, Jim DiCarlo here at MIT has been doing very elegant experiments in monkeys where, while they're doing visual recognition, you have one of these models and you ask, can I predict the activity of neurons in different parts of visual cortex from this computational model? And to the extent that you can do that, then you think that the model is approximating not only at the behavior level, but also at the physiological level.
Give you the idea of where to go record? Or does it just become useless? So if you've already matched electrophysiological data, I assume the architecture will be different. It'll be a different model. So what do you do with that model?
Ideally, the model can give you predictions about where, how, about the experiments, and about how to go about doing this type of matching. I think many of us, including Jim and us and many others, think that there's some sort of virtuous loop where the model can suggest experiments, and then we can record neurophysiological data. The neurophysiological data can show us where the models are wrong, and the behavioral data as well. I think we need to match both, right? Behavioral and neurophysiological data can suggest where the models are wrong, or what aspects of the model we still need to explain, and we can go back and forth between these three levels of analysis: behavior, physiology, and computation. I don't think we can get away without behavior. I think just understanding a system at the neuron level is not enough. I think understanding behavior by itself is not enough. I'm particularly excited about the intersection of the three, about being able to do neurons, behavior, and computation.
Closed loop kind of between the two. And it seemed like one of your experiments was a small example of that with this generative process and then this single neuron. What do you think is kind of the future of those kinds of experiments? They seem to be more common and more accessible now with new technology. Like, what do you think? What do you think those might reveal, or do you think that will become like a big paradigm in the future?
Yeah, I think there's going to be more and more of that. I think there's a lot of things that we can do in silicon that neurophysiological experiments are very challenging. They take a lot of time. They're very expensive in a lot of ways. We get a lot of information from them, but there are a lot of things that we can first try in silicon, for example, in a computational model, and then have informed hypotheses about how to explore the neurophysiological system. There may be a lot of situations where we, in real time, we may be able to fit models with neuronal activity, train those models and improve those models in real time, and then suggest what's the next best question. A lot of the way we do experiments right now is sort of trial and error, right? So we say, well, let's throw a handful of pictures. Let's throw this picture and this picture and see what happens. And we record activity, and we hope that there will be a difference. And lo and behold, we have been able to see differences in that way. But what if we ask, what's the most informative question that I can ask now? Maybe the most relevant question to constrain my model is to actually show this picture like this and like that, or I don't know, some other. So the model can suggest which gradients or which directions are more interesting to explore. Yes, I suspect that there's going to be a lot of, there are already, and there will be even more fascinating interactions between physiology and computation. All right. If there are no further questions, thanks a lot.